All right. Welcome, everybody. So, this is a very short introduction to population genetics theory, and we will essentially cover two key concepts. The first one is the concept of Hardy-Weinberg equilibrium, and the second one is the concept of linkage disequilibrium. And as you will see in the following lectures, those two concepts are central and instrumental for you to understand a lot of the statistical methodologies underlying genome-wide association studies and their applications for prediction, for example, using techniques like polygenic risk scores. But let's not get ahead of ourselves and start from the beginning. What is population genetics theory? So, population genetics theory is concerned with characterizing and quantifying genetic variation that exists within and between populations. And as you see here, I've highlighted genetic variation in populations because those are two important concepts that we'll also need to define. So what is a population? Well, I wanted to start from the natural way anyone will think about defining a concept like that is to turn to the dictionary. And so if we do so, we come across various definitions of a population, and I've highlighted four of them here. So the first one from the Oxford Dictionary defines a population as all inhabitants of a particular place. The second one, which again from the Oxford Dictionary, but has another put more emphasis on the biology, and it defines a community, the population as a community of animals, plants, or humans, among whose members interbreeding occurs. Essentially, that reproduction can occur between those individuals, which is an interesting, which add another layer. It's not just a special notion, but there's also this notion that some genetic material can be exchanged. Let's go to a third one, which is from the Australian Bureau of Statistics, which defines a population as any complete group, okay, with at least one characteristic in common. So, which suddenly makes it very broad. And finally, I guess this is probably my favorite because it's very comprehensive, is the definition provided by the World Health Organization. And so this definition emphasizes that it's a set of all inhabitants of a country, a territory, or a geographical area, uh, and total or for a given sex or age group at a specific point in time. And in demographic terms, it is the total number of inhabitants of a given sex or age group that actually live within the border limits of the country, territory, etc. But let's try to highlight what all those definitions have in common. I guess they have a notion of space, partly because, well, populations or individuals tend to live congregate in small spaces, so that there is this notion. And I guess there is this interesting notion brought by the Oxford Dictionary about interbreeding. So there is a notion that, which is a part that for a population genetics lecture we will be also interested in. So we're thinking about interbreeding happening. And maybe what is the very specific and important aspect that this fourth definition is bringing is the notion of time, because space and interbreeding defines a population, but the notion of time is also quite critical. Which leads me to think about a population almost as droplets. And this is an analogy that I like to use because if you looked at the three drops that have been highlighted and ask, are those droplets from the same population? Well, the answer is it depends. It depends on your time scale. When exactly have you measured those droplets? If you could go back in time, you probably see that they will be very close, close next to each other. And also there is this notion of space in the x-axis. And I think from all that, just wanting to highlight that defining a population is a bit of an abstraction, and that wanting to see this idea with you, that this is going to be a model. You're going to think about a population as a mean for you to answer a specific question. But depending on the context, the way you've defined a population may not always be the best way. So keep that in mind. It's a little bit of a concrete abstraction, and which means that who is inside or outside of the population can be arbitrary and will depend on your research question. But let's just for the sake of crystallizing some ideas, think about it as it's a group of individuals. And here, if you think about the humans, a group of humans that can obviously, because we're part of the same species, can in principle interbreed, and that they may or may not have this notion of spatial congregation, but that's something that may depend on the type of question you may want to ask. So now let's move to the second part of that definition, which is about genetic variation. And here as well, there are some challenges because if you compare the DNA sequences of two individuals, there's near infinite numbers of ways you can measure similarity or dissimilarity between those two sequences. But the way you typically do that, at least not to get lost into the infinite spaces, we usually use reference genomes, and we will essentially look at a single reference for multiple people. So which means that from the perspective of that reference, we can identify individuals who will have a variation relative to that reference. So that variation can be measured in terms of the number of repeats, some specific motifs in the DNA sequence that are repeated. It could be inversion. A particular motif is inverted in some individuals and not in others. It could be deletion. So that particular part of DNA is present in the reference but is absent in some individuals. Or, and that's going to be the most common way for us to think about genetic variation. We're going to look at a very localized variation at the single nucleotide or single letter in that sequence. And that's what is illustrated on the right-hand side of this slide. So here we have three individuals, and you can see around that particular position of the sequence, uh, the sequence is exactly the same, but at that particular position, we can see that first individual has an A, the other one has a G, and the other one has a T. And this kind of variation is often called single nucleotide polymorphism or SNP. Just to very briefly, before going back to our definition again, we're going to define an allele as a possible state of DNA sequence at a particular locus. Locus will be a way for us to define a position on the genome. And so here, if we go back to that particular variation, we see that people can either be A, can either be G, or can either be T at that particular position or locus, and that will define the allele. So we can have the A allele, the G allele, or the T allele. And because I guess in humans, we inherit our DNA material from mom and dad, at each position, the sequence can have two possible states depending on what was the state of that position in mom or in dad. And the state of all alleles at a given locus is going to be called the genotype. And so here, going back to that particular position, because we have three possible states, we can have a lot more genotypes. We can have individuals being AA, we can have individuals being GT or TT, and those will be called homozygous or homozygote genotypes, or individuals will be called homozygous. And those one will be when the two states are the same, the ones inherited from mom and the one inherited from dad, or they can be heterozygote. So the genotype is an heterozygote state, or that individual at that particular position is heterozygous when the alleles inherited from mom and dad will be in different states. So for example, one is in an A state and the other one is in the G state. That gives us the AG genotype, or one is in the G state and the other one is in the T state, and that gives us the GT genotype. So just to end on this first part, then if you think about rephrasing what population genetic theory is, is going to be a theory of alleles and genotypes frequencies, because essentially replace the notion of variation by some more statistical terms. We're going to look at the frequency of those alleles and genotypes and how those frequencies will vary within and between groups of individuals that we're going to broadly refer to as populations. And so in the next part of this lecture, we're going to introduce the notion of Hardy-Weinberg equilibrium and what drives changes in allele and genotypes frequencies, and all the way down to talking about linkage disequilibrium. So thanks for watching this first part, and I'll catch you up in the second part.